All right, let's get right into this because we are on the edge of a genuine biological revolution. I'm talking about a project that's as ambitious as the Human Genome Project, but this time it's for all of life. It's called the Biodiversity Cell Atlas, and its goal is nothing short of creating a dictionary for the book of life itself. So think about this for a second. Over the last few decades, scientists have made some absolutely incredible progress. Thanks to massive efforts like the Earth Biogenome Project, we've managed to sequence the complete genetic blueprints, the genomes, of more than 10,000 different complex species. That is a truly monumental library of life's source code.
But here's the catch, and it's a big one. Having that entire library isn't the same as being able to read any of the books. As this quote gets at, we have all the letters, all the words, but we really struggle to understand what they do. How does that sequence actually build a living, breathing organism? There's this huge gap between the code on the page and the life in front of us. And this huge problem has a name. It's called the genome to phenotype gap. We have the full text, the entire book for a species, but we just can't connect the genes, the words and sentences, to the actual story they're supposed to be telling. That story being, the shape, the behavior, the entire life of that organism. So to put it as simply as possible, having thousands of genomes is like owning a library filled with books written in a language you just don't understand. To make any sense of it at all, what we desperately need is a Rosetta Stone, right? A comprehensive dictionary that can translate that genetic code into actual biological function. And that dictionary is exactly what the Biodiversity Cell Atlas, or BCA for short, is all about. It's this massive, coordinated, international effort designed to finally let us start reading that incredible library of life that we've spent decades collecting. So what is it really? Well, the mission is crystal clear. It's about bringing together two huge fields, biodiversity genomics and single-cell biology. The idea is to create these incredibly detailed molecular maps, or atlases, for a massive range of species. And, this is key, to build them all on shared standards so we can actually compare them and start uncovering life's most fundamental rules. Now, the whole project is built on this really simple but totally profound truth. Every single cell in an organism is constantly reading the genome. So if we can study those individual cells, we can essentially eavesdrop on that process. We can see exactly which genes are being used, when they're being used, and where. It's brilliant. The process itself is pretty straightforward, at least in concept, though it's super challenging in practice. Basically, scientists take a sample from an organism, they very carefully separate it into individual cells, and then they use these amazing single-cell sequencing technologies to read which genes are switched on in each one. Then, powerful computers take all that data, cluster it together to identify all the different cell types, and ultimately build this beautiful, detailed atlas for that organism. Now, if you want to understand just how big of a deal this could be, think about AlphaFold. The AI that basically solved the protein folding problem. That was only possible because it had a massive, high-quality database of protein structures to learn from. Well, the BCI is aiming to create that same kind of treasure trove of data, but for decoded genomes. This is the stuff that will power the next generation of AI to finally bridge that gap between genotype and phenotype. Okay, so let's say they pull it off. Once we have this dictionary, this unbelievable data set, what can we actually do with it? Well, this is where it gets really exciting, because the potential applications are just transformative. They span pretty much every single field of biology. I mean, just look at biotech and medicine. The possibilities are just staggering. We could discover brand new enzymes for industrial use, or find new medicines, like antimicrobial peptides, hiding in the genomes of some obscure species. Once we truly understand how genes work together, we could even start to design synthetic life, actually creating genetic circuits from scratch to perform specific tasks. Wild stuff. And for basic research? Oh, it's an absolute goldmine. We are almost guaranteed to discover cell types we never even knew existed. But bigger than that, we can finally start tackling some of the huge questions in evolution, like how did neurons first evolve? We can do that by comparing these cell maps across totally different animal groups. It would literally let us build a family tree, not of species, but of the very cell types that make them up. The impact on environmental science is just as huge. We could finally study the vast majority of microscopic life, that 99% that we can't actually grow in a lab. Or think about coral reefs. We could untangle the complex cellular dance between the coral and its symbiotic algae, which could be the key to fighting coral bleaching.
This is going to lead to incredibly powerful new tools for monitoring the health of entire ecosystems and for guiding conservation. Of course, an ambition this grand doesn't come without some immense challenges. I mean, this is a journey into completely unknown biology, and scientists are going to have to overcome some pretty significant hurdles if they're going to map the entire tree of life. So the project is tackling this with a really smart, phased approach. We're in phase 0 right now, and this is all about getting the methods right. It's about developing and standardizing all the experimental and computational tools, basically building the toolbox. Once that foundation is rock solid, phase 1 will kick off, and that's when they'll scale up data production across hundreds and hundreds of species. And the hurdles really fall into 2 big buckets. On the experimental side, think about it. How on earth do you gently get single cells out of a plant with a tough cell wall, or a tiny little insect, without stressing them out so much that they change which genes are turned on? Then, on the computational side, how do you take all this messy data from thousands of different species, analyzed with different technologies, and make it all speak the same language so you can compare it? These are the really tough problems the BCA is working on right now. But as these challenges get solved one by one, the BCA is poised to kick off a completely new era for biology. This is going to be an era where we can finally connect that digital code of the genome to the living, breathing, functioning world that it creates. And the ultimate vision here, it's just breathtaking. It isn't just about making a collection of individual atlases. No. The real goal is to connect them all into a single unified framework, a cell-type tree of life that can trace the evolutionary history of every single cell type, all the way back to its ancient origins. This project truly promises to change our understanding of evolution and of life itself, and it leaves us with a really exciting question to think about. Once we can finally read the entire book of life, what incredible new stories will we be able to write?